In Chapter 12, we're going to talk about biotechnology and genomics. So biotechnology is really just talking about using living organisms or their parts to create products or to learn more about biology. One of the most exciting aspects of biotechnology is genetic engineering, and this is where scientists can insert clone genes into an organism. Once that organism receives a clone gene, it's referred to as a transgenic organism, meaning that it has genes from different species. So when we talk about gene cloning, it's really just the process of making identical copies of a specific gene. We mentioned the genetic code previously that shows the 20 different amino acids that we have. The genetic code is almost the same across all organisms. So because of that, it makes it possible for us to move genes between even very different species. And this is really important because it helps open up potential for improving medicine and agriculture and a lot of other things. So a big part of biotechnology is recombinant DNA technology or rDNA technology. And this is where DNA from two or more different organisms is combined to create something new. So to get that foreign DNA into a host organism, scientists have to use a vector. And a vector is just a carrier. Usually it's a plasmid, and a plasmid is just a small piece of circular DNA that bacteria often have. So in order to cut that DNA at specific spots, it's going to require the use of something called restriction enzymes. And when we do cut it with the restriction enzymes, it leaves behind something called sticky ends that can easily stick to other DNA pieces. The final enzyme needed is DNA ligase, and this is going to come in and help seal all of those sticky ends together so that we can create the recombinant DNA. So one of the first applications of recombinant DNA technology was actually in the production of human insulin, and it is still used today. So before genetic engineering, insulin had to be harvested from animal pancreases. This was a very slow and expensive process. But now scientists can take the gene for human insulin, they can clone it, and then put it into a plasmid, and then insert it into a bacterial cell, and then use the bacteria to produce the insulin as if it were their own protein. So this makes the hormone much more affordable and widely available for people with diabetes. So this is just showing you the process. Here's the human insulin gene here. We can clone it. So here's the insulin gene here. Here's our plasmid, which is going to represent our vector that bacterial cells have. We're going to cut it at specific sites using a restriction enzyme. Then we are going to insert the insulin gene into this plasmid. We're going to add DNA ligase to help seal those sticky ends together. And what we produce is recombinant DNA. So then the recombinant DNA can go into a bacterial host cell. Bacteria replicate and multiply very, very quickly. And as they do this, they're producing huge amounts of insulin, or the particular protein that we want to. So another important application in biotechnology is the polymerase chain reaction, or PCR. So PCR is a technique that's used to help amplify or to make lots of copies of a specific DNA segment, which is really helpful if you need to analyze a small sample of DNA. So we have an enzyme called DNA polymerase, and this is coming from a bacterium called Thermus aquaticus. And this enzyme is going to be responsible for copying the DNA, even at the high temperatures. Because this DNA polymerase is coming from Thermus aquaticus, this is a heat-loving bacterium, so it won't denature and it won't stop working at these high temperature levels. PCR does require the use of primers, and these are just short pieces of DNA that target a specific section on the genome, and it's making sure that only the region that you're interested in actually gets amplified. The PCR process involves repeated cycles of heating and cooling, and it's going to be doubling the amount of DNA each time. So it makes it a lot easier to study, especially again if we're starting with a very small sample of DNA. So, when it comes to DNA analysis, one of the most useful techniques is DNA fingerprinting. And this method takes advantage of the fact that certain parts of our DNA, called non coding regions, are different for each person. And these regions have repeating DNA sequences, and the number of repeats varies from individual to individual. So, scientists can use PCR to copy these regions and then run the DNA through gel electrophoresis. And that's just a process that helps separate DNA fragments based on their size. So the longer pieces of DNA move more slowly through the gel, while the shorter ones are going to move faster. And what it does is that it's going to create a unique pattern. And this pattern is really like a genetic fingerprint that can be used to help identify individuals or even help establish biological relationships. So, biotechnology and genomics, they're changing the game in several fields from medicine to forensics. And tools like genetic engineering, recombinant DNA, PCR, and DNA analysis really make it possible to manipulate and study DNAs that were once impossible.